Hello everybody. In this video, we'll look at question number 21 from GATE Biotechnology 2022 paper. So the question is, in animal cells, the endogenously produced microRNAs silence gene expression by option A, base pairing with 3' untranslated region of specific mRNAs, option B, blocking mRNA synthesis, option C, binding to the operator site, option D, base pairing with a 3' region of specific rRNAs. To answer our question, let's learn about RNA interference. It is a term given to the process of RNA silencing in the animal kingdom. RNA interference or RNAi is a mechanism that inhibits gene expression at the stage of translation or by hindering the transcription of specific genes. RNA interference targets include RNA from viruses and transposons. For example, when we are infected with a virus, the genetic material of the virus is most usually RNAs. So these RNAs are recognized by the cellular machinery and are targeted so that they are rendered functional and do not affect the host cells. There are several components and steps involved in RNA interference. mRNA silencing is achieved with small interfering RNAs abbreviated as siRNAs or microRNAs abbreviated as miRNAs that form a complex called RISC, which is the RNA-induced silencing complex. This cleaves the mRNA. So with respect to small interfering RNAs, the double-stranded RNAs are processed by proteins called dicer, and the single-stranded antisense small interfering RNAs associated with other proteins involved in the RISC complex. So RNA silencing could also happen with microRNAs. So the hairpin-like primary microRNA is processed by a protein called DROSHA to form a pre-miRNAs. This is then processed by proteins called dicers to form single-stranded mature microRNA. This then associates with the RISC complex. These RNA molecules in the complex acts as guide for the RISC to associated with complementary single-stranded mRNAs. RISC then binds to the mature mRNAs at the 3' UTR. So once the RISC complex binds to the 3' untranslated region of the mRNA, it results in the degradation of the RNA or results in the inhibition of protein synthesis or translation. Either way, the RNA becomes non-functional. So this is the mechanism of RNA silencing or RNA interference. Now coming back to our question, in animal cells, the endogenously produced microRNAs silence gene expression by. So like we saw, the microRNAs silence gene expression by binding to the 3' untranslated region of specific mRNAs. So the answer here would be option A, base pairing with the 3' untranslated region of specific mRNAs. Because option B is blocking mRNA synthesis. The microRNAs they either degrade the mRNA or they block the translation of proteins from these mRNAs, and they don't bind to the operator site and they don't bind to any rRNAs. rRNAs are not involved in the whole process. So the answer here is option A. Thank you.